So our next speaker is Carol Adair from the University of Vermont, and she is going to talk about connecting leaf and root decomposition to soil carbon.
So this will be a little bit of a change from the last presentation, but perhaps harkening back to Esmerette's talk as well as the one about root decomposition that came after her this morning. And this is work that I did several years ago at UC Merced on my sabbatical with Esmerette and Dave Rice and some other folks there at UC Merced. So this is talking about mostly litter decomposition and kind of what litter, root and leaf litter turns into while it's decomposing. Is one of the big debates in ecology and biogeochemistry is what is soil carbon and where does it come from and what is it made out of? So to do this work, I drew on some archived samples from the long-term intersite decomposition team experiment, or LIDET, dataset, which was a 10-year decomposition study. It was spatially extensive. I'll show you where those sites were in just a moment. And it used a really wide range of litter quality substrates. So this experiment decomposed litter type, a whole variety of litter types at 27 sites from the Arctic all the way down to the tropics. These are the sites here on this side, and you can see that these sites covered a really wide range of mean annual precipitation and mean annual temperature. So a wide range of sites, although sort of all in North and Central America. It used a litter bag decomposition study. For folks who aren't familiar with that, basically you take a known amount of dried litter, leaves or roots, and stick them in these mesh bags that look like this, and put them out in the environment to decompose, and you collect them or collect some of them over time to see how much has disappeared over time. So this experiment used 6 standard leaf litters and 3 standard root litters, which means that they got the leaves and the roots from one place and took them to all the sites to decompose. So that's a standard leaf litter. So the same leaves, the same roots at all of the sites. And then they invited researchers at the different sites to decompose their favorite litter types. And these were called wildcard litter types, and they were decomposed at only 1 or 2 sites. So there were 4 replicates per site. This is what a replicate looked like. So one replicate was 10 sets of 10 litter bags. So one set had the 9 standard litter types, leaves and roots, and 1 wildcard litter. And then 1 set per replicate was collected once a year for 10 years, or more frequently at tropical sites because everything just goes so much faster there. So that is kind of the basic background, except for this part. They used a really wide range in initial litter chemistries. So this is showing you percent nitrogen on the y-axis for the leaf and root litters, and percent lignin on the x-axis. And just to give you kind of an idea of what these are, these 2 over here are kind of poor quality, right? High lignin, low nitrogen content. So this is slash pine leaves and roots. And then these 2 are sort of on the tastier end of the spectrum for microbes. So this is Drapetes glauca. It's a tropical species, very high nitrogen content and low lignin content. And Pacific dogwood is another one with pretty high nitrogen content but very low lignin content. So as a little bit more background, we did some work with all of these data. So all of these data are available online, and you can look them up and see how much mass was lost over time. And we did some modeling work and found that a pretty simple model explained about 70% of the variation in the LIDET data set based only on initial litter chemistry and climate, so site climate variables. Using this sort of a 3-pool model with fast decomposition based on how much nitrogen was in the litter, slow decomposition based on the amount of cellulose that was measured initially, and then very slow decomposition based on the initial amount of lignin. And this was all characterized not with NMR, for example, but using kind of methods. This all happened in the 90s. So using sort of wet chemistry methods. So this is just showing you some of the results, hopefully showing you that we did a pretty good job of explaining litter decomposition over time. So this is a low quality pine litter with low nitrogen, high lignin, and that tropical high nitrogen, low lignin litter type. And hopefully the model, so the data are the means with the standard error bars, and then the model that we came up with are the solid or dashed lines right here. So pretty decent job there. But one of the big, so we can sort of predict how much is left, but we really don't know anything about what this stuff is, what is actually left after it has decomposed for 10 years. So that model that I was just showing you assumes that it's lignin. So this is a sort of dated view of litter decomposition where we don't really account for any transformation. We just assume that lignin decomposes really slowly, so that's what's left. And this sort of historical view of litter decomposition is sort of codified, I don't know, not codified, but shown in this figure here, which is after a Swift decomposition textbook showing that as litter decomposes over time, what's left are leaf parts, right? And the most slowly decomposing parts are what's left after a long time. So this is sort of an older view of what happens to things as they decompose, leaves and roots. Sort of a more emerging view is the idea that microbes can eat everything, right? They eat lignin, they eat cellulose, hemicellulose, and they turn it all into microbe stuff, microbial stuff, right? Either dead microbes or other microbial products. So that leaves and roots that are decomposing shouldn't be old plant parts, right? It should be, or it should have turned into microbial products. So that is the emerging view. And the question then is, what actually happens? Okay, so along with that, does what happens, does what remains in that leaf or root litter depend on if it's a root or a leaf? So some of the literature suggests that yes, it does. And this is to remind me to tell you that the roots actually were decomposed below ground. So the leaves were decomposed on the surface and the roots were decomposed in litter bags below the surface. And the literature that's out there suggests that, or seems to kind of argues that roots decompose faster and are turned into microbial products more quickly. So the idea here being that maybe leaves don't really turn into microbial stuff that well, but maybe roots really do. Another thought is, does it depend on if litter is high or low quality? So there is some argument in the literature saying that low quality stuff doesn't really turn into microbial stuff very easily. Whereas if it's high, if the litter is high nitrogen, the microbes really like that. They'll eat it up and leave a lot of microbial stuff. So the idea then that high quality litter turns more into microbial stuff than low quality litter. So that might look something like this, right? With low quality litter looking more like plant parts, high quality litter looking more like microbial parts. Okay, so what did we do? With NMR, we couldn't analyze all of the samples because it's a huge number of samples. So we took some of the archived samples from four very different sites. So this site up here in the Arctic, Harvard Forest, which is a temperate deciduous forest, Central Plains Experimental Research Station, which is a short grass steppe or a dry or arid grassland, and then Loquio Biological Station, which is a wet or moist tropical forest. So we used two litter types. We used that high quality Drapetes roots and leaves, and then we used low quality conifer roots and leaves. And I was lucky enough to be able to use some of the archived samples to analyze, and we looked at the initial samples are time 0, 1 year, 3 year, 5 year, and 10-year archived samples. So this is what I brought with me, 71 samples in total. That's a lot for NMR, I'm told. To Merced and worked with Dave on their 13-seat NMR solid state Agilent machine. And the idea then was to characterize what's in those samples and how it changes over time. So once we got the spectra from the NMR, we integrated these spectral regions here, which you may be familiar with. And then we also used sample carbon and nitrogen content and put all of that information into a molecular mixing model from Baldock et al. 2004 to infer these biomolecular constituents that ecologists like myself find it easier to think about. So things like carbohydrates, protein, lignin, lipids, and carbonyl. So we did that for all of the samples. But first, I just wanted to show you that what some of those spectra look like. So the litter that I'm showing you here is a high nitrogen, so that Drapetes leaf, in a temperate forest site. So that's that Harvard forest site. Over here, this is showing you in orange the initial leaf and the 10-year leaf in blue here, and the initial root here in red and the 10-year root in blue here. So some changes that you can kind of just see from looking at this include, oh, there's my text, an increase in the alkyl carbon as the leaf decomposed, right, from the initial to the 10-year, a decrease in O-alkyl carbon like cellulose and hemicellulose, a little bit of shifting around in the lignin or aromatic C carbon area, and then some changes perhaps in the carboxyl region. And we can see these same changes kind of mirrored in the root spectra over here. Okay. So returning to our questions, thinking about does what is remaining depend on if it's a root or a leaf, or if it is high or low quality? So these are some figures based on some of the ratios that you can develop from those integrated regions. So this is the O-alkyl over methoxyl ratio. So it's sort of like holocellulose over lignin. And we can see as things decompose more or less across the board, we get a decrease in holocellulose relative to lignin with decomposition. We also looked at the alkyl to O-alkyl ratio here, which is the ratio of sort of lipids, waxes, and cutin to cellulose and hemicellulose. And what we see is an increase, generally speaking, not always, in the waxes, lipids, and cutins relative to cellulose and hemicellulose with decomposition. So we saw that more change below ground. So with the roots that again were decomposed below ground, but also a little bit more in the high quality litter than in the conifer leaf litter. Okay, so then this is moving to some of those mixing model results. So here I'm showing you carbohydrates as a proportion of whatever is left over. And in general, we see carbohydrates decreasing over time, right? And in all cases, but not at the same rates. And then lignin sometimes decreasing, but often staying just sort of hanging out and staying around, which maybe was a little surprising, but nevertheless, we saw more change again below ground in general, not always, but in general, and often more change in the high quality litter, again, especially not always with the lignin. Okay, then we also are looking here at the protein content, right? as a proportion of the mass remaining. High quality here, conifer over here. And again, we're seeing sort of an increase overall in the amount of protein and often an increase in the amount of lipids, especially in the below ground cases, but also somewhat in the high quality versus the low quality litters. Okay, so these figures are kind of combining all of that information into single figures, and I'll show you these again. So this won't be the only time you'll see them, but here we have lignin in blue, carbohydrates in yellow, lipids in brown, protein is green, and the carbonyl is gray. There's very little carbonyl. It's all sort of at the top there. And the thing I think to see from these figures, so this is all versus percent mass remaining. So percent mass remaining being sort of an indicator of how decomposed is it, right? So if you're in the Arctic, this looks like it's cut off because things just didn't decompose that much in the Arctic, right? Whereas if you are in the tropical forest, you got a lot further down towards 20% remaining or even less mass remaining. So I think the things to see here in general are that we got these big increases in lipids, some increases in proteins, especially below ground. And then again, a bit more change kind of within each of these environments in the high quality than in the low quality litters. Okay, so going back to sort of our theoretical construct here, we saw more change below ground in roots, sort of turning more into microbial stuff, those lipids and proteins, and maybe a bit more change in the high quality versus the low quality litter towards microbial products and away from plant products. Okay, so what is remaining, we found also depends largely, as you might have guessed from some of the previous slides, on mass remaining, but not on time. So these are PCA results looking at, so it just put in the PCA is just looking at the mixing model components, right? So here we have protein and lipids if you're headed this way, lignin and carbonyl and carbohydrates heading this way. And this is really organized much more by how much mass is remaining, right? Moving from not very decomposed. So this is 100% mass remaining down to very little mass remaining when you're in sort of this protein and lipid area. If we do that same, this is the same PCA, but now it's colored by time and years, you can see the times just sort of everywhere. So it makes more sense to think about what this stuff turns into as a function of how decomposed is it rather than how long has it been out in the environment. And that's largely due to the fact that we used these such different environments to decompose them in, from the Arctic down to the tropics. Okay, so it depends less on site, which was a bit surprising to me. I sort of expected to see the Arctic all circled up towards the undecomposed stuff, but really all the sites are everywhere. So the Arctic are these circles, for example, and those circles are everywhere. So things didn't really cluster by site, surprisingly, but they did cluster a little bit by species, right? So the conifer roots and leaves are orange and yellow, so you can see them clustered here and over here, whereas the high quality stuff, purple and blue, kind of turning into this more microbial, like lipids and proteins and stuff over here. We're still thinking about this, so if anybody wants to talk about it, I'd be happy to do that. Okay, so thinking about our theoretical framework. So I've talked about all of these components, but I haven't really tried to put them onto our framework. So let's try to do that. So here, this is a figure really based heavily on a Terry Chapin et al. Textbook where he has sort of the composition of litter over time and uses these different time axes depending on what environment you're in. So the Arctic here going from 0 to 100 years, right? Because things take a really long time to decompose in the Arctic. The temperate, well, the tropics is the other one that he has in his textbook, going from 0 to 3 years, right? Because things decompose very quickly in the tropics. And then some friends of we some friends of mine and I, or colleagues, I should say friends, some colleagues of mine and I decided that temperate systems like the short grass or the dry grassland and the temperate deciduous forest, maybe we'll put that on about 30 years. So that is the center axis here. So lignin is here in blue, proteins plus lipids being sort of microbial stuff in brown here, and then the carbohydrate in yellow. And you can see it kind of following this figure here in general. But another way to think about this is, as I just said, everything seemed to be organized by mass remaining more than time. So as to put it on this figure. So this figure is a little bit tricky to think about because mass loss here, right? So we're losing mass. This is less mass remaining over here. But to make this figure, we have to have mass remaining on both axes. So it's a little bit tricky to interpret this figure in exactly the right way. If people have ideas about how to do this in the best way, I'd love to hear that. So I think that it's useful to look at this figure, but it's also really useful to think about these components also sort of as a proportion of the mass remaining, right? So it doesn't matter in this figure over here, you've got mass remaining, right? 100, this is all the mass remaining, and then this is less and less, right? So this is increasing mass loss as you move in this direction, but you're always summing to 1, right? So you can see more clearly that the lipids increase over time and that the proteins increase over time and the carbohydrates decrease over time. So this, again, this is the high quality DRGL or Drapetes roots. So we saw the most change in this particular litter type. This is the high quality leaves. So less decomposition over here in both of these cases. A little bit less change than we saw in the roots, but still some increase in the lipids and the proteins, and decreases in the carbohydrates. Again, lignin really hanging on as it did in the roots. Conifer roots showing maybe not as much change as the high quality stuff. So again, we have kind of the component remaining as a percent of the mass remaining over time, and then as a function of mass loss. And again, sort of the proportional values of each of those components. So it's a little bit easier to see what's increasing over time. Not as much change, again, in the conifer roots as in the previous two. And then conifer leaves really showing the least amount of change and least amount of decomposition overall. We look here, you can really see how things did not change very much for those conifer leaves. So kind of some take-home messages here that what is left of leaves and roots really depends on how much is left or how decomposed is it versus simple time. Whether it's a root or a leaf, right? We saw more change in the carbon components in roots than we did in leaves, but we also saw a bit more change in the high quality litter than in the low quality conifer litters. So the theory, I showed you these sort of the historical view and this emerging view, and what we found was really somewhere in the middle, right? It's not all plant parts, and it's not all getting transformed into microbial stuff, right? Plant components are persisting for a pretty darn long time. 10 years is a while, especially in deciduous environments. So there was still lignin and there was still even carbohydrates hanging out. But we did see this shift towards lipids and proteins. thinking of that as more microbial products and microbial stuff. All right, with that, there are a lot of people to thank. Here's Dave and the postdoc that Todd that worked with us at UC Merced, and the uh, NMR that I was always afraid was I was going to hurt. Here's Mark Harmon, who was kind enough to share his archived litter samples with us for this work. So thanks to everyone, and I can take any questions if there are questions.
Thank you, Carol. And for this must have been, I remember during COVID time.
Yes, I know we all have masks on, right?
So, questions? Yes.
Sorry, missed. Curious, how much of a perturbation, or is there a lot of discussion about whether there is a perturbation from the bags? Like, is decomposition different if things are just allowed to sort of slowly seep into the soil instead of being in a bag?
Yeah, absolutely. So actually, one of the things that I think would be really interesting to look at here is that they did do an initial for the first couple years, a study where they had kind of the small mesh size that these bags I'm talking about today used versus one that allowed soil mesofauna to get in. So things like nematodes and termites, and they have really big impact. So I think a really interesting question is, do those things have an impact on what the litter turns into? And I don't think that I don't know. We're working with the UVM NMR person who dropped off the bottom of this slide, Monica, to look at some of those samples just to see if there might be some interesting things to think about there. But I think in general, yes. So if you just have your litter sitting out, the litter bags do have small impacts on microclimate. So, hanging onto water, maybe sometimes being a bit warmer, but also what they let in and what they exclude. So definitely worth thinking about all those things.
Thank you.
Yeah.
Third question.
Yeah, sorry, I came in a little bit late, so maybe this was discussed earlier, but it kind of tails off from the previous question. You're looking at lipid and protein growth here.
Mhm.
And you ascribed that to microbial, but was there any fungal component to this? And maybe that's the reason why the lignin persists, since lignin is typically eaten by things like white rot.
Yeah, yeah. They can definitely get into the bags. So there can be fungi in there. And actually, that is a question that I would have for anyone who is better at NMR than I am, is that like, what does that look like? What do fungi look like in an NMR, right? Like, do they look like any of these components? Does anyone know?
I would guess, I'll just guess, but that they're pretty much, that all living things are pretty much the same when you break them down into
Yeah,
lipids, proteins, and I mean, the animals versus plants.
yeah,
But
yep,
that's just my guess.
yeah.
Put anything in a solid state NMR spectrometer and it looks the same.
Nice.
I actually,
But so yeah, fungi could get in, but I don't know what the signature of fungi would look like versus a microbial signature, or if they just look the same in an NMR.
that is something to look in more carefully.
Yeah.
More questions? Yes.
Can you answer the question all our neighbors have about whether or not we need to clean up our leaves or we can let them lay on our lawns? Which is better?
Well, no, I can't. I mean, as somebody who loves decomposition, please leave them there. And who likes to rake unless you like to jump in the piles of leaves? Um, I mean, it's hard to grow your grass if you let the leaves go there. But yeah, I don't have an answer for you.
Well, thank you, Carol.
Yeah.
And let's give her a hand.